Hello, everybody. We are the Los Angeles Waterkeeper Practicum team. Our advisor is Levi Simons, and we are very proud to present the work that we've been doing for the past year. Our amazing team is shown here. The overall goal of our project is to promote science that may lead to better protections for freshwater stream quality across Los Angeles. Currently, 98% of streams in Southern California's urban areas show signs of degradation to ecosystem health, meaning that the streams aren't supporting a healthy community structure. The need to understand the health of our existing water bodies is critical for any future restoration or anti-degradation efforts. Currently, stream health is assessed in the Los Angeles River Basin using specific pollutant levels and physical qualities, such as pH levels, the presence of toxic chemicals, or sediment disturbance. In a vacuum, these analytes provide a limited view of whether a freshwater stream can be considered healthy for an ecosystem. By looking at the biological communities that can thrive in these areas, we have a much fuller understanding of how multiple pollutants interact to whether a stream is healthy for the organisms that live in it and the people that live near it. We hope that through our project, we can better understand water stream health and preserve the health of our ecosystems. The California Stream Condition Index, or CSCI, is the tool that was used to score the biological health of freshwater streams. Benthic macroinvertebrates are small creatures found in freshwater bodies, existing in a wide variety and abundance. Many species are particularly sensitive to chemical and physical changes in water quality. Therefore, by analyzing the presence or absence of species in the macroinvertebrate community, we can develop a holistic picture of stream health. A CSCI score closer to 0 represents an impaired or degraded water body, and a score greater than 1 to a peak of 1.4 indicates a healthy reference site. Our research question was, does this particular measure of biological health of streams, the CSCI, act as a reliable indicator of water quality across the watersheds of the Los Angeles region? Breaking down our goals into two parts, we first aim to determine, using machine learning, the degree to which the CSCI could capture variations in regional water quality of LA watersheds, and subsequently to produce a research paper based on our findings and submit this to a peer-reviewed journal. Once published, we hope that these peer-reviewed findings can be used by LA Waterkeeper to encourage the implementation of biological indicators in water quality assessments for Los Angeles. We focused on the Los Angeles jurisdiction of California's State Water Resources Control Board, designated Region 4. The boundaries can be seen in the image to the left. The regional control boards set statewide and regional policy regarding water bodies. And to the right is the map of the hydrology across the LA region.
Machine learning refers to algorithms that improve automatically through experience and the use of data. And this can be applied to environmental data to test the success of different water quality indexes. For the bulk of our analysis, we used a type of machine learning called random forest to assess if current sample data is sufficient to assess biotic integrity on a regional scale. We utilize both occurrence and environmental data to both model the data graphically as well as create maps to visualize the data. For our purposes, we input a set number of water quality samples and saw if random forest could accurately predict other CSCI scores for watersheds within the region. We built and evaluated an initial set of 1,000 random forest models of the CSCI as a function of watershed, sampling year, and various chemical analytes taken from the sample data for the region. These samples were taken over a 20-year span with a total of 685 samples. The source data for each model was obtained by randomly selecting 180 samples from our study area, which is what we found to be the minimum number needed to consistently model significant predictions for the CSCI. Each of these sample groups were split into training and testing sets for the random forest model to predict CSCI scores for the watersheds within the region. What we found to be the top 9 most important chemical analyte variables were selected to create the set of models along with their individual partial response curves. These curves plot the CSCI scores against the concentrations of the analytes and also show the occurrence of the scores within the region. And on the right of this slide are the partial response curves, which are arranged in order of most important in the top left corner to least important in the bottom right. Ultimately, we found the CSCI to be reliable. Even with the differences in the CSCI scores within the watershed studied, it captured 72% of all variation in water quality across the region. And so with this, we found strong evidence of a measure of biotic integrity acting as a reliable indicator of chemical water quality.
We found the mean correlation between our initial set of modeled and actual CSCI score to be highly correlated, as shown by the correlation coefficient of R equals 0.73 and a standard deviation of 0.16. However, since most of the variables in our initial models are of low relative importance, the performance of our models changed only slightly when based solely on the 9 most important variables, in which the correlation then became R equals 0.74 with a standard deviation of 0.16. With this study, we would also like to discuss possible limitations to our analysis. First, while over 200 unique chemical analytes were used in our data, our model of the CSCI was effectively described by using only 9 of those. This reduction in model complexity was possibly due to the low frequency with which most analytes were measured. As a result, this low sampling frequency for a majority of the analytes limits their potential to contribute to our models of the CSCI. However, the 9 analytes of interest in our model have been observed to influence the biotic integrity of communities across a variety of freshwater stream ecosystems. Another limitation is that some analytes, such as phosphorus, had concentration distributions strongly skewed towards low values. As a result, this skew may reduce our ability to better model the CSCI response to changes in phosphorus concentration. In the meantime, our team hopes to educate and increase awareness by sharing our results and having a draft of our freshwater manuscript ready for review. We will continue this dialogue in consumable ways for the public by sharing information through our website. Additionally, we have been and will continue to communicate with Los Angeles Waterkeeper to discuss what steps we will be taking moving forward, as well as what steps they can take to implement these practices involving biological indicators. Thank you all for your time.